So, as I told you, we are going to discuss about genetic engineering today. So, what is genetic engineering? So, here some of those points are mentioned, but overall, I will cover like the overall idea about genetic engineering. Say, for example, I told you that, like all of you know actually, that E. coli cell, like if this is an E. coli, and it has one chromosome, right? Circular chromosome. And this chromosome makes all the product 11 for E. coli cell itself. But now, if you are interested to put some of your interest, that means some gene, you are interested on that gene. But that gene is not part of E. coli itself. So somehow currently we have very good technique. So you put your gene of interest here, right? So basically what you did, manipulation of an organism's genome. So the E. coli chromosome is E. coli genome, and now you put some of your interest here, some gene of interest. So as a result of that, you manipulate the organism's genome. And then it can range from changing one base pair, deleting a whole region of DNA, or introducing an additional copy of the gene. So for example, already you have this E. coli cell, right, that contain E. coli chromosome in addition to your gene of interest. So for example, gene A, you already inserted here. Now, you are not happy with your wild type, that means what is naturally available, this gene A. If you believe that gene A makes insulin in human, okay, just I'm giving example, that gene A, when it present in human, it makes insulin. All of you know that whatever DNA or gene present, from there you are getting mRNA, and from mRNA you are getting protein. Insulin is a protein hormone that helps in glucose metabolism. So now, when you make this E. coli cell and then you actually make your protein, you will get insulin. And there are a lot of technique available in the lab so that you can actually isolate insulin from this E. coli cell. But you are not even you are interest on some more thing. For example, you want to change some of the amino acid present in insulin so that the insulin will be more active or it will be more useful. That is also called genetic engineering, because you are not changing those amino acid after purifying the protein. Rather, you have to change some codon or some nucleotide sequence in the gene A itself. So as a result of that, you will get modified version of insulin, maybe that is more useful, right? So those are the main, point of genetic engineering. And the last point here you can see, used to enhance or modify the characteristics of an individual organism. So this is little bit more about, so I will discuss this point at the almost at the end of this presentation or at end of this today's class.
Sir,
Yes.
there was like this rice which contained vitamin A, because
Hmm.
Asian countries have deficiency. Like it is called yellow rice,
Yeah.
and is it also a product of genetic engineering?
Yeah. Yeah. Exactly. Exactly. So, so the rice they produce vitamin A, right? So the golden rice is that is like mostly golden rice, right?
Yeah, 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 yeah.
Yeah, golden rice. So not only that, actually one of our Indian scientists mostly like work on that golden rice. Yeah, you are right. So I will give you different areas of example first, and then I will discuss how what are the basic techniques to do that. So let us discuss. So here, So an overview of genetic engineering, as Soham you just told about golden rice. I didn't include golden rice here, but some other examples are there. So, for example, agricultural application, and this one you might be aware about this BT cotton. Sometime you will get some news which are a little bit disturbing that because of BT cotton, there are a lot of problem in for farmers in Maharashtra, Telangana, all those region. But when BT cotton was made, that was with good intention, but it has some, I would say, a lot of controversy there. But apart from, I'm not going to discuss that part, I would say, but I would like to discuss the scientific part here, that how genetic engineering actually help in agricultural application. So, BT cotton. So, any one of you are aware about BT cotton without Google search? Just if you are aware, because most of you are actually, too young to know that, because mostly
Sir,
around.
I only know that it is some kind of pest resistant type of
Yeah,
thing.
you are right.
It
So
doesn't get destroyed that
mostly,
easily.
like when you were like most of you, almost um, uh, like I would say almost 20 years back, actually, that is not your age then. So during that time, actually, it has been started in India. So let's see. Uh, BT cotton, this is cotton plant. So that you understood that this is cotton plant. Now what happened? BT, this like later came from one bacteria name actually uh, that is called Bacillus thuringiensis. If you forget this name, nothing will happen. But only I would like to say this B and this T, Bacillus thuringiensis. This is a bacteria. Now, this bacteria naturally they produce one toxin, right? That is called, because of this Bacillus thuringiensis, this is called BT toxin. This BT toxin is very much harmful for some, insect, mostly like bollworm, some kind of moth. So those like insect, they are very much harmful for cotton plant and they destroy cotton plant. As a result of that, what scientists did, they transfer this gene. So BT toxin is a protein I already introduced, but it is coming from one gene, right? So they somehow transferred this gene where? In the cotton plant. So as a result of that, like leaf, stem, almost all part of this cotton plant will make the BT toxin. So I think you understood, right? The cotton plant itself, they are making this BT toxin in their leaf. So as a result of that, now those bollworm or whatever those insect, they are actually eating those cotton plant, they will die. So as a result of that, these BT cotton are now insect resistant. So you don't need to apply regular chemical insecticide. Rather, this cotton plant, they are actually making some product in their cells so that they are toxic to those worm. And this is, I would say, good that way, that you don't need to provide chemical insecticide again and again. But one drawback is, all like this is some problem with some, big company, because they actually retain some of those information and they engineered in such a way that you have to buy those seeds and you are planting, right? But whatever the cotton plant you are getting, next generation, those cotton plants are not BT resistant anymore. Right, that's the problem. That's the problem with the farmer. That means you have to buy those seed again and again every year from like with some like good amount of money, and that is a little bit problem for poor farmer, right? So like that if you search in Google, you will get a lot of news in Indian national newspaper and many other sources. I'm not going into that, but I hope that you understood the overall concept. This is BT cotton, and in India now, most of the cotton actually cultivated is BT cotton, and in US, China, everywhere, most of the cotton producing country, they make BT cotton in today's world.
Sir, excuse me.
Yes.
Sir, that means you said that the seeds of BT cotton are means they cannot germinate to another plant.
Yeah. So you see, this is Monsanto and Indian company Maheko. They work together. So this is the major problem. Say, for example, I'm just giving you one example. Whatever capsicum you get in your market, right? Good quality capsicum. If you use their seed, mostly you will see that that is not giving the similar variety. So that's problem actually. But this is little bit of thing. But what I told that is like purely designed in such a way that you have to buy those seeds. So neither they will germinate or they have some problem like even I don't know, because that is normally retained those information with the company itself. So if that get germinated, but they will not be resistant to those worm, right? So let me come back again in the slide. And apart from that, genetically modified sweet corn. So I'm not going into details about this sweet corn, but this is mostly, in US, they cultivate this, mostly in Western world, they cultivate this genetically modified sweet corn. So that has high sugar content, and that sugar is used for corn syrup and so many other purposes. Application in dairy, poultry farms, all of you know that growth hormones to increase the production of cow milk. So from where you will get this growth hormone? Again, this is product of genetic engineering, and also in poultry and many other, this kind of industry. And here, again, I would like to emphasize here that environmental application, it might be a little bit new to you, that uh, application of oil-eating bacteria. So this Pseudomonas putida, so this is some kind of bacteria. So in general, bacteria, they actually, they cannot utilize hydrocarbon like oil, petroleum. They cannot use the carbon source from those petroleum, right, or other kind of oil. But somehow scientists engineered this bacteria. The natural counterpart was there actually. Some bacteria were like that. They have special ability. But from there, scientists actually improved and they generated some oil-eating bacteria. So I hope that you are enough intelligent that to understand what will be its use, oil-eating bacteria, right? If somewhere like oil spillage or some environmental cleaning is required, then you can pour this oil-eating bacteria so that they can utilize those oil as their energy source. So basically, they will metabolize those things, and that will help to clean the environment if it is required. And it has been applied in multiple, places. Okay. And next. So particularly, I would like to mention one thing here, this oil-eating bacteria, one of the major scientists who work on this and contributed huge amount of finding here. His name is Dr. Ananda Mohan Chakraborty. So he is actually Indian and he studied in Kolkata University. And when he he was working with this bacteria, oil-eating bacteria. At that time, he was working in GE. Like GE company, right? So he was in GE, and later on, I forget actually what he did, but during that time, like almost a long time back, and he actually contributed a lot in understanding and developing this oil-eating bacteria. And the last one here, I'm mentioning the therapeutic application. This is very important one, that human insulin. So people who have a lot of diabetes, that diabetes cannot be controlled by diet, or some other restriction or exercise, then they need to take insulin. Like they need to take insulin in a routine basis. Those insulin, like all those insulins are getting produced through genetic engineering approach, right? Here I'm just taking one screenshot from Amazon, that even you can buy this this reagent to control pests, particularly mosquitoes. So this is again very similar to whatever I told, that Bt toxin, like Bt cotton. In the similar way, actually, they are selling this. This is a commercial name. They like make it mosquitoes. So this is people are nowadays using to control mosquito. Okay, so just you can search and you can read about it if you are interested. That's all. Now, 2-3 more example I will like discuss, and then I will go into the details that how we can make it. So here, this flavor saver, this is a genetically modified tomato. So nowadays, I would say most of the tomato you are getting in like big market, supermarket, those tomato are hybrid tomato, that you know, right? But most of them, many of them, they are this kind of tomato, flavor saver. So how people actually like make this tomato, but what is the purpose? Because if you actually I'm not sure whether you are aware, if it is not hybrid tomato or not this kind of tomato, those tomato are a little bit more sour, as well as their wall is more thin. So as a result of that, it is very difficult to transport those tomatoes. Say for example, you are transporting tomato from one place to another place, and it is in truck for 2-3 days. When it is actually getting transported, many of those tomato they will get destroyed. Okay, because of that they can ripe very quickly, and they have very thin wall. So I'm just showing you here. If you see here, their wall is that outer layer is actually very thick, and they are very tight even when they are already completely ripened. So as a result of that, this is very good, so that you can store for a long time in supermarket as well as at your home, but nothing will happen. But normal like desi tomato, whatever you say, those are not that good. You cannot keep in, I'm not telling about the food quality. Remember it. Food quality might be better in those tomato which are not engineered version. But this is again, we are in the in a situation where this commercial and all those things are also important, right? So as a result of that way this is useful, that people can keep, even you can take some of this tomato and keep at home for 1 month, even more than that, nothing will happen, right? So this is like just one example, and the next example is glowfish. So here you can see this is called zebrafish. Whatever you can see here, they are zebrafish because they have stripe like black and white stripe. So zebrafish. This is the wild type or normal zebrafish, and now people actually scientists they introduce some proteins. Say for example GFP. This is called green fluorescent protein in some fish, and they will glow as they will look like this green. And there are many other similar version of protein. So for example, YFP or yellow fluorescent protein. This is present in this fish, and RFP red fluorescent protein that is present in this fish. So now these fish are commercially available, and people are keeping in aquarium also, right? But their original counterfeit part is looks like this. This is also good looking, I would say, but you can make something like this. Those are called glowfish. And now this is the last example I'm going to discuss, but mostly here you have more information, not just a kind of statement. So that production of human insulin, as I told you, that insulin is a very important hormone, and it is absolutely required in our body so that you can metabolize your glucose. And all of you know when you are eating food, right? So finally you are getting glucose, but how those glucose will be utilized, right? You have to use those glucose in order to get energy. Energy means here ATP, right? Adenosine triphosphate mostly, right? So that means those glucose should go inside your cell, and inside your cell there are like some metabolic reaction you studied in your class 8, class 9 level, that glycolysis, Krebs cycle, all those things that will give you energy. But before getting energy, you have to transport glucose inside the cell. And do you know who does this job? Insulin. Insulin actually helps to transport glucose from blood to cell, or from outside to inside of the cell. That is the function of glucose in our body. Okay. So now, if you see here that this is the best example of genetic engineering, or this is also called that recombinant DNA technology. And nowadays, as I told you, insulin is getting produced through this method. And I would also say, or I would like to state here, India is a huge like production house of insulin. Even India actually export insulin in other, like, other countries. Not today, it's for a long time actually. So we have good like technology to make insulin, although this is not a very kind of very like you don't need that much of sophisticated thing, but it is standardized very much. So let's see how insulins are getting generated in our body. So here, insulin, as I told you, this is nothing but a polypeptide. This is called protein hormone or polypeptide hormone. So you can tell, like, last week I told you that in a protein you have the N terminus, you have the C terminus, right? And this is the polypeptide chain. This is called pre-pro-insulin. That means insulins are just getting produced in your body, and now that insulin should be actually secreted from the cell. Do you know where insulin is getting produced in our body? Any of you? Without
Pancreas.
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 Yeah, very good.
So islets of Langerhans in pancreas.
Yes, so inside the pancreas you have some, like, in pancreas is an organ present in our body, and pancreas contain a lot of different types of tissues or cells. Like, tissues means many cells together, we say tissue. So there are some cells are there, those are called islets of Langerhans beta cells, and those cells actually make insulin, but that needs to be secreted from the cell, because only pancreas is producing insulin, but you need insulin everywhere in your body. So as a result of that, that pancreas needs to secrete insulin in your blood, right? And that should travel everywhere. As a result of that, insulin will be available everywhere, and that can actually help, right? So here you can see this protein or polypeptide, they contain some signal sequence. That signal sequence means this will not be part of the mature protein, but it will help to secrete from the cell. Those details you can forget, nothing will happen, but I'm trying to explain you, this has some relevance to day-to-day life, so I'm just trying to explain those things. So because of proteolysis, this signal sequence will be like cleaved, and this is almost the final version of insulin. Here you can also see some disulfide bond formation here. Last week I discussed what is disulfide bond, and this is called pro-insulin, and this is not yet mature insulin. But interestingly, you will see, although you have read before that splicing is happening in mRNA, but here also the similar thing you will get. We don't say this is splicing in this case, or in case of protein. This is some kind of cleavage. So you will see that one portion of this protein is not required in mature protein, in mature insulin. This portion should be actually cleaved. So this should go away from this like mature insulin. And now This is the mature insulin, and here you can see this insulin has the activity. It can help to transport glucose from outside the cell to inside the cell. Here, if you see properly, you will find that now you have two polypeptide chains. See, one polypeptide chain here and another polypeptide chain, and they are attached through disulfide linkage, and they have some kind of three-dimensional configuration also. Right? We are not showing it here, but you can see this C peptide that get released, but this is your mature insulin. This is how insulins are produced inside our body, inside our cell. What I want to say, those processes are very much complex, but we can make this version of insulin in our lab very effectively and efficiently, this insulin, and that is being produced in a routine basis to provide insulin supply for a patient who needs to take insulin in a routine basis. Any question? If you have any question, I will like to address that, and then I will go to the next slide.
Hello, sir. Sir, for genetically modified eatables like fruits and vegetables, how do they take care that it does not cause any other side effects like cancer or all?
Yeah, that is some problem actually. You are right, but mostly most of these genetically modified things, whatever I believe, this is just my personal opinion. I'm telling you, I believe like even today's world, whatever the chemicals we use for insecticide and for many other purposes to keep those things fresh and it's looking better, those things have more adverse effect than this kind of genetic engineering. To me, most of this genetically engineered version of this food, they might not create any problem during our metabolism processes. Rather, I would say, like when you guys actually buy, like for example, I'm just telling you that brinjal means bengan, eggplant. Those are highly, almost like every week, every day, a farmer used to put some insecticide, right? So those like chemical, they have, I would say, more side effect or direct effect to make this kind of problem. So Rajesh, is it clear? But those concerns are also there. If you just search whatever you ask, those concerns are also there. That I am not disagreeing with that.
Sir.
So the best way is in order to address your question, the best way is neither of these are good, right? So you can most of the traditional things are better, but the problem is you have to match with supply and the demand chain. So that is also important. So that is another way you can think. Now. Here, the insulin is an indispensable hormone in our body. I already discussed a little bit about insulin. Here you can see that this is the pancreas, and from pancreas, insulin is getting generated, and insulin binds to insulin receptor, which are present in our cell. And whenever insulin binds to insulin receptor is another protein. This is when the interaction is happening, then glucose get transported inside the cell, and that provides energy. But the thing is, this is absolutely required, because whenever you are actually eating something, say for example you are eating some rice or some roti, then what will happen? Like after digestion, like the amount of glucose is increasing in your blood. That is called, if increase level of glucose, that is called blood sugar, right? But in a normal person, in a healthy person, what will happen? The insulin will be more. Whenever you are taking more and more food, the insulin production will also get increased. So as a result of that, higher level of insulin will help to transport those glucose from blood to cell. and that will be instantly metabolized and give you some energy. But the thing is, we have like disease like diabetes. So in case of diabetes, then somehow this insulin production or the normal function of insulin is somehow hampered. So in case of type 1 diabetes, this is autoimmune diabetes. In this case, the cells which are producing insulin, those cells are getting destroyed by our own immune system. That's why called autoimmune diabetes or type 1 diabetes. I will discuss this in more details in the immunology class. And then another category of diabetes is called type 2 diabetes, which is more prevalent in India. So mostly in Indian population, you will not get type 1 diabetes. Mostly type 1 diabetes available found mostly, I would say, Norway, Sweden, mostly those Western world and like in those countries. But in type 2 diabetes, this is kind of normal, I would say, metabolic disorder, and this is very prevalent in India. So here what happened, although insulins are produced, their level is low, like low level of insulin is getting produced. And in addition to that, some kind of resistance happened, so that although you have insulin, but still it is not helping to metabolize glucose. So at the very beginning or early stage, you can control your diet and you can do some more exercise in order to take care of it. But in some cases, even without restricting diet, after restricting diet as well as exercise, still it is not under control. So in that case, that person needs to take insulin in a routine basis. So I'm not going through it, whatever written here, but I already.
So, why is the insulin production reduced in type 2 diabetes?
Why you are telling, right?
Yes, sir. In type 1, it was destroyed by the immunity, but what is happening in type 2?
Yeah, good question. But I would say even no one knows properly actually. Like as I told you that one thing is insulin production is less, and the other portion is like even many people they have seen that even insulin production there, but somehow insulin is not getting recognized, or they are not interacting with the insulin binding receptor actually, which are present on the cell surface. So as a result of that, what happened? Although you have insulin, but glucose metabolism is not taking place. Okay, so in order to answer your question, I would say it's not very much clear actually what is happening. But if you ask some like diabetologist, some medical person, I'm sure that will be able to answer like at least highlight some of those things. But at my level, I don't have that specific answer. That only thing I would say, if someone do proper exercise and all other things, that might help actually.
Yes, and sir, one more question.
Yeah.
So in type 2 diabetes, sometimes the patients, when they glucose level is over the glucose.
I think Kavil Hari Krishnan, I can see only that person, right? So something happened. Just a minute. Oh yeah, now it's fine. So can you ask question again?
Yes, sir. That in some type 2 diabetes people, they give something called insulin shots. So if the receptors don't work well, then even taking insulin shots wouldn't
Very
help.
good. Very good point. Like this is ideal question. The way I was explaining, I should get this question. You are very good. So you are right. But see, this working is not, I would say, yes or no kind of thing. But they need, if you increase the amount of insulin concentration, then some of those molecules will still bind. So that way you are helping actually, right? So like insulin receptors are there, they're not like that, they are completely inactive. If they are completely inactive, then it is a kind of you cannot control anything, right? That means how you will get energy, like the glucose will never be metabolized, not like that, but their functionality get reduced. The only thing is how you can achieve this function, you are increasing the insulin concentration. So although they cannot bind to insulin receptor properly, but if you increase the concentration, then the chance is more that more and more molecule will be attached to the insulin receptor, and that will transport the glucose. Tanmay, it is clear?
Yes, sir. So basically, the probability of attaching we are
Yeah,
increasing.
because this year actually we didn't discuss enzyme kinetics. So there we actually discussed this thing like competitive inhibition and other kind of enzymatic reaction that will help you to understand. But you are right. This is not yes or no. In that case, if insulin receptors all are gone, they cannot work. Then you cannot treat that people at all, right? How you will control that, right?
Yes, sir.
That is
Got
not
it.
possible.
Yes, sir. Got
And
it.
now here,
Excuse me, sir.
yeah.
Sir, is there a type of diabetes where lot of insulin molecules are produced?
Can you ask
Eh,
this question again?
Insulin is produced in excessive amount. Is there a type of diabetes like that?
Yeah, if insulin get produced in excess amount, you are saying.
Yes, sir.
Yeah, so this insulin production is highly controlled in our body, right? So mostly, like for example, when you are sleeping, right? So for example, at 9 p.m. you took your dinner and you are sleeping, and morning at 7 p.m. again you wake up and you are taking some breakfast. During this time period, so we have very good control mechanism so that insulin will not be produced in huge amount. Otherwise, that if the blood glucose level is like dropping too much, that is also dangerous, right? So as a result of that, in that slide is written actually, Rajas. So the insulin production is coordinated with the when you are eating something like and that time you need to like metabolize sugar, that means glucose. That time the production will be high, and that is the problem actually. When you are taking insulin from outside, then you need some control mechanism. I will discuss a little bit about that also. Okay, but that way, like it normally, even say for example fasting blood sugar. What is your fasting blood sugar? When you take your dinner during say 8 p.m. or 9 p.m. at morning, if you test normally in a healthy person, what should be sugar level? Say around normally the value you get is around say 80, 75, 85, some concentration actually, some amount per milliliter of your blood, right? So that is normal because it should not drop like much beyond that. So if you are not taking your food, but I would say still we have some stored carbohydrate in our body that can be used to provide glucose. Okay, so that way we are very much controlled, so that will not be a big problem. So here you can see the effect of insulin. Just whatever in the previous figure, it's in a small place is written. Here you can see in more details that some insulin receptor are on present on this cell membrane. So this is basically cell membrane. Okay, cell membrane, and this is the cytoplasm, that means inside the cell. This is insulin receptor. Whenever insulin bind here, this insulin binding will help to make more of this transporter on this membrane, right? So this is called glucose transporter, like GLUT4, glucose transporter 4. So whenever this and like insulin will bind to insulin receptor, you will see that more and more glucose transporter are functioning on the membrane, and the function of glucose transporter is nothing but to send or to taking glucose from outside to inside. And now this glucose molecule will be metabolized and it will give you energy. And as a result of that, the amount of glucose in the blood outside the cell will also get decreased, and which is required. Okay. And now this slide again, I would say this enzyme like GLUT4, all those things, if you forget, nothing will happen. I'm just trying to explain you the overall idea. The production of insulin, like in long time back in 1921, Frederick Banting and Charles Best, you have studied in your school days most likely. They purified insulin from dog's pancreas, and over the years, scientists made continuous improvement in order to produce insulin. But the problem is, see, uh, if you are taking insulin from outside, like from dog's pancreas, from horse pancreas, pig's pancreas, and you are injecting to human body, then the problem is you are actually injecting some foreign protein, and that sometimes causes some immunogenic reaction in those persons, and that is not a good way of handling this scenario. Also, how much insulin you will get from dog or from horse, that is also challenging. So first time in 1980, actually scientists, they made insulin in the lab. And in 1982, one big pharmaceutical company, many of you are aware about this, Eli Lilly. So it's a big pharmaceutical company. They made insulin, completely human insulin from genetically engineered, through genetically engineered way. And at present, researchers could produce genetically engineered insulin in an unlimited supply without depending on animals. Then the question is how we can make this. So they are in order to answer this question, I will come into this steps required in genetic engineering. So whatever I introduce, that is some of those day-to-day information. Say for example, as I told you, even some tomato, some mosquito, killing those solution, as well as this insulin. But let's see how people make this thing, particularly for insulin, right? So we will discuss that in details. Keywords in genetic engineering. From here, some of the important thing you have to understand. The first thing is gene of interest, right? So these are keyword, means gene of interest means, say for example, you are interested on some biological project and you are working in my lab, and I told you, okay, make this protein and we would like to test its functional or its enzymatic activity, like some particular protein. Then that protein should be encoded by some particular gene. If it's a human protein, then it's from human, some of the human gene, right? So that particular gene is now your gene of interest, from where you are getting the product, that is gene of interest. Now, the next thing is, those things don't forget, okay, gene of interest. Now, plasmid. Plasmid is very important, okay, never forget this, plasmid. So plasmid is a small circular double-stranded DNA molecule that is distinct from cell's chromosome. So I would like to say, in some bacteria, so they have bacterial chromosome, circular chromosome, right? Double-stranded chromosome. In addition to that, they have some small DNA element. Those are also double-stranded, small DNA like circular here. So these are plasmid, okay? Plasmid. Not all bacteria, they have plasmid. Some bacteria in natural condition, they have this plasmid. And what is the function of those plasmid? Most of the time, they provide some antibiotic resistance, okay? Antibiotic resistance to those bacteria. So they help those bacteria to survive in some condition where you have some antibiotic solution, right? This is called antibiotic resistance. Yeah.
So in many days ago, there was a disease, tuberculosis, and they actually talked about stage 1, stage 2. So they talked about antibiotic resistance. In the first stage, the antibiotic resistance was not developed much, but later on it developed more. So was this due to the plasmid?
No, Tanmay. That is not because of plasmid. So this whatever I am telling you, this is in natural condition. Even I would say some bacteria which are present in soil, in some organisms, some cattle. Sometimes they have this antibiotic resistant gene in normal condition. But Tanmay, what you are telling, antibiotic resistance, this is very important question. Although I should clarify a little bit. Not in great details, because then it will take one class. So what is happening? Most of the mycobacteria, like for TB particularly, like people used to take a lot of antibiotics. They need to take in order to for the treatment. Now those mycobacteria, they little bit mutate themselves, their genome get mutated because of those drugs. And what happened, some of those, like, pump means some transporter present on their cell wall, like on the bacterial, on the membrane, you can say also, like they have some transporter, somehow they effectively transport those drugs from inside to outside. So as a result of that, you are taking antibiotic drug, but the problem is whenever that drug is going inside the bacteria in order to kill those bacteria, as I told you that many antibiotic, they stop translation, many other things. But problem will be they have good, like, good devices. Like, for example, I'm giving you just one example, like some transporter protein that is called efflux pump, that will efflux, or that will send out those, those drugs to outside again. So as a result of that, those bacteria are now antibiotic resistant. This is just, I'm giving you one mechanism, but there are a lot of other mechanisms that way bacteria can be antibiotic resistant. But Tanmay, this is not like what I am telling you, that is not the case in case of mycobacteria. Okay.
Okay, sir, got it.
So here, so I told about plasmid, and this vector means some subtle, like, vector means, I would say in this case, the plasmid itself also an example of vector. That means at the very beginning today, I introduced that you have to, like, like send your gene of interest to E. coli, right, in order to get your protein. But the thing is how you will transport your gene of interest. The idea is vector. Vector means you studied vector, like, that mosquito is also called vector because mosquito carries some disease, right? But in this case, vector means we are telling some DNA element which can carry the foreign genetic material or gene of interest. So for example, plasmid double-stranded DNA, it is also, you can use plasmid as a vector. That means you can put some gene of interest inside the plasmid, and that plasmid you will finally send it to some bacteria, and then your gene of interest will be expressed in the bacteria and you will get the product. Some of the thing you might not understand, or you cannot, like, connect all those things, but I promise you that after some time, even like if you are enough, if you are providing enough attention, within 30 minutes you will see, you will get all those links. I will discuss all the way so that you can connect everything together, that how we make a foreign protein inside E. coli. Okay.
Sir,
And
I have one question.
Yeah.
Sir, does the plasmid like participate in transcription?
Participatory transcription. Yeah, very good question. So when I will discuss a little bit more about the plasmid, I have slide also. You are right, because whatever the gene you put in the plasmid, that needs to be transcribed, right? So that is your question.
Yes, sir. Otherwise, it won't be
Yeah,
expressed.
exactly. Yeah, yes, it needs to be transcribed. It needs to be translated. I will go into that. Okay. So yeah, I will discuss that. So now, if I, if I tell here, so those like few keywords, those are absolutely important. Gene of interest, plasmid, vector, plasmid, they are overlapping. Like I would say, like some plasmids are vector, but apart from plasmid, there are some other vectors. I am not going into those details. It's not required. Now, the transformation. Transformation means you are aware about transformation. In your first class, I discussed about the Griffiths experiment, that those like rough strain of bacteria, they become smooth strain because they have taken some genetic material, some DNA, like from the smooth cell of bacteria. I think some of you can remember, right? So this
Yes,
is
sir.
called, yeah, this is called transformation, that you are actually, like some bacteria, they are taking some genetic information from outside source and they are getting transformed. And this process is called transformation. So as a result of that, what we will do in my lab, I have different types of plasmid. Now also we have not just naturally available plasmid, we have some engineered version of plasmid so that we can work in a better way. So I have many plasmid in my lab. So that plasmid, one of those plasmid, I can use to, like, make some engineering. That means, as I told you that some of you are interested, you are working in my lab and you are actually taking some gene from some human genome, right? And that gene you are putting into this plasmid, and that plasmid, this plasmid is containing the gene of interest now. And this plasmid you can use to transform bacteria. That means some bacteria, for example, some E. coli strain present in our lab, they don't have any plasmid. They have only the bacterial chromosome. Now you can put this plasmid that contain your gene of interest, which you are working on. So that plasmid you can transfer into those bacteria. That is called transformation. Now your bacteria, they have their own genome, that is called bacterial chromosome. Also, they have this plasmid that contain your gene of interest. Now you have to use this bacteria. You can grow this bacteria in large volume, say for example 1 liter of media, 5 liters of media. So as a result of that, you will get huge amount of proteins which are coming from those gene of interest, and those proteins will be accumulated inside E. coli cell, and finally you have to extract and harvest those protein, and you can work on that. That is the overall bottom line, overall I would say the outline, what you need to do. Okay, now let's see how we can put gene of interest inside the plasmid. And one point you cannot see here is written as host cell. Host cell means those E. coli here, that where you will transform, right? Mostly like E. coli cell we use, but apart from E. coli, there are many other things, like even you can express your protein some other sources, but that is more complex. I'm not going into that details. Okay.
Excuse me, sir.
Yeah.
Sir, the vectors, are they necessary to be a DNA molecule, or they can be any organism too, like bacteriophages?
Yeah, so as bacteriophages, in order to answer question your answer, I would say yes or no, because the thing is bacteriophages, but they are also like that way. We are again using the bacteriophage DNA. Okay, so that way. But here in the genetic engineering approach, whatever we are saying that plasmid or vector, that is DNA, right? So now the question is whether it is plasmid or any other DNA. For example, as you told, bacteriophage DNA, that can be also used, and people use that. Okay, but that is more complex. That will be difficult to explain how we can use bacteriophage DNA. But here in this case, in case of genetic engineering, vector means DNA, and there plasmid is one classic example, and I will stick to plasmid only.
Okay, sir.
Now, here, this is one important slide, but I would say this slide, you don't need to go through it. I will sum up this slide in this way. Let's see. Then you will get almost all the information available. Here. So, this is a plasmid, double-stranded DNA, okay? And you are interested on some gene, say gene A. So that gene A is present in some human cell. So how you will get human cell? Yeah, you can get some like little bit of human cell from many sources. In lab, we have some human cell line. That means we actually store that cell line in liquid nitrogen, like at very low temperature, so that in use, when we need, we can use some of those cell line. It can be some kind of specific body cell, right? And in culture, that means you can grow this cell in proper condition. I'm not going into the details, otherwise, we will go into completely different direction. But the idea is, from this human cell line, you have to isolate the DNA, okay, the whole DNA. And now, from this DNA, you have to particularly take the gene A, how you can take this? You have to do some kind of PCR, you know, that polymerase chain reaction. So as a result of that, you will actually amplify this gene A. You have many copies of gene A, and now you can work on this gene A, and you have to insert this gene A into plasmid. Now, some of you might have some question that, you are using DNA, but some of those protein, like, some gene, they might have intron, exon. How you can take care of this? Then intron will be here, and then that intron, splicing will not be taking place inside the E. coli cell. If you can actually link those things, that is good. But I would like to say that this is very simplest way I'm telling you that directly from DNA to, we are making the PCR product, not like that. Actually, we do some more steps in between in order to take the particular, portion of the gene which are already processed. That means you don't need to, you will not face the problem of exon, intron, anything. The only thing is you will get the gene of interest and you have to put that into plasmid. So as a result of that, now your plasmid looks like this. So you have your gene of interest here, gene A, and this is your plasmid, okay? Now, the idea is this plasmid, you transfer it, that is called transformation, in one E. coli cell, okay? So this is E. coli chromosome and this is plasmid, okay? And all these plasmid, they contain gene of interest A. And this E. coli cell, you can grow in some media, some vessel, like some flask, you are growing E. coli. Now I'm drawing E. coli like small like this. Those are E. coli cells. But the thing is, this plasmid, they have some property. One of those important property is they have some antibiotic resistance gene. So for example, this plasmid, they have some antibiotic resistance genes. That means say ampicillin. Ampicillin is an antibiotic, ampicillin positive, ampicillin resistant gene is present here. So as a result of that, what happened? If you put this bacteria in this solution, in the media, in order to grow bacteria, in this medium, bacteria can be doubled in every 20 minutes, as I told you before, and you will get millions of bacteria. But now, if you are keeping some antibiotic here, ampicillin. Ampicillin also present at particular concentration in the media. Then what will happen? Then bacteria wants to survive in presence of ampicillin. Normal bacteria, if they don't have plasmid, they will not survive. Only those bacteria, they have this plasmid, they will survive. Do you know why? Because this plasmid contain this antibiotic resistance gene. This portion of this plasmid, they can make some product, and because of that product, those bacteria are resistant to ampicillin. Ampicillin is an antibiotic. But if you increase the ampicillin concentration, then what bacteria will do? Bacteria will try to survive. Then the thing is, plasmid will be increased inside the E. coli cell. The number of plasmid will increase a lot. You know why? Because all of this plasmid, they contain one copy of this resistance gene, ampicillin resistance gene. If you have more concentration of ampicillin in the solution, in the media, in order to survive, then bacteria will try to make more and more antibiotic resistance product, which will give it to antibiotic resistance. So how they will do that? They need to express those genes, right? Antibiotic resistant genes. But one of the important property of plasmid is that plasmid can replicate independent of the bacterial chromosome replication. That means I told you before that in E. coli, you have one bacterial chromosome. Whenever the bacterial chromosome, they get double, that means after replication, that bacteria will also divide into two. From one bacteria, you are getting two bacteria, right? But this plasmid, they can be replicated autonomously without depending on the bacterial chromosome. And that is an advantage. That's why we are using plasmid. Remember here, I am not sure whether you can connect it or not. Why? What is the importance of antibiotic here? Whenever the transformation happened, for example, consider that one E. coli cell that contain only one plasmid, and that plasmid contain your gene of interest, gene A. But because of this antibiotic selection pressure, bacteria wants to survive and it increases its plasmid number inside the cell. This plasmid number can go even up to 500,000 per cell, even more than that. 500 to 1000 per cell, right? So while this plasmid is getting replicated inside the cell, don't forget that your gene of interest is also getting replicated. That means 1000 plasmid present in that cell means 1000 gene of interest, that gene A is also present in each of those E. coli cell, right? That is a huge thing. In your body, in your cell, that gene A is not present in 1000 times. It's present only twice. I told you, right? That two chromosome, like each chromosome you get like, one pair you get from your father, one pair you get from your mother. So only twice this information present. But in E. coli cell, you have 1000 times this present. It's an arbitrary number. It can be even more. So as a result of that, now if you have all other things in place, as actually Soham asked that whether the plasmid can be transcribed or not, yes, I discussed about transcription before, that if you put your gene of interest, that gene A in particular place where you have the promoter, operator, all those things during lab operon I discussed, right? These are a little bit different, but you have to clone in such a way so that inside the bacteria, this gene A or gene of interest will be transcribed into RNA, and that RNA will be translated into protein. As a result of that, inside the E. coli cell, you will get huge amount of your protein or the product which you are interested, right? That is the idea. So this is like, overall like discussion what we are doing and what we want to do. And only thing, few specific step I will discuss in this whatever present in the slide, that how you can put this gene of A, the gene of interest, into plasmid. That is a good question, because if you can imagine, or if you can try to figure out the dimension of DNA, like what's the width, what's the length between two base pair, you cannot use normal engineering equipment like whatever we normally use in your lab. So for example, scissor, scalpel, or nothing like that you can use, right? So that means we have some special tool to precisely cut DNA in between nucleotide sequence, and that should be in our control, and then you can transfer that in a particular portion, particular DNA. So that part I will discuss. So now tell me, whatever I discussed last 5-6 minutes, is it clear to you?
Sir, I have a question.
Yeah.
Sir, you said like when we like isolate the gene A from the DNA, from human DNA, say, we use PCR. But the thing is, like in PCR, the promoter sequence won't be replicated, right? Like like there is a promoter sequence to which the A binds, and it won't be replicated, right? But when we insert the gene A into the plasmid, if the promoter sequence is not present, how will it be transcribed?
Yeah, so Soham, actually we normally take just gene of interest. That means whatever the corresponding to protein sequence, only that portion we will take, and we will put in plasmid. And in the plasmid is already engineered, and that those plasmid we use in lab. So in the plasmid you have those promoter, operator, all those side. Why? Because anyway, like inside the eukaryotic cell, we have different type of RNA polymerase, but we are using bacteria as a protein production unit here. So we have to use our bacterial RNA polymerase in order to transcribe, right? So as a result of that, for the time being, you just consider that we are taking the corresponding portion of the gene from human, which makes this protein only, nothing else. And other things is present in the plasmid itself, so that in plasmid, after the promoter, you have to put your gene of interest. Okay, so as a result of that, this promoter will invite bacterial RNA polymerase, and that will transcribe our gene of interest. But polymerase, RNA polymerase present in our body, that is different. Is it clear, Soham?
Yes, sir, I got it.
Yeah. So now I will discuss 15 more minutes. Then I will give you some break, because after that discussion will be a little bit shifted. Then we will discuss about the cloning of the whole organism, like animal itself. Okay. So just bear with me another 15 minutes, and then I will be done for this portion. Okay. Tools for making a collage. So here quickly I will go through it. This is some analogy that DNA of interest I told you before. Scissor, you cannot use scissor in order to cut precisely nucleic acid. You have to use restriction enzyme. This is one enzyme, particular enzyme. After the discovery of restriction enzyme, then only we started genetic engineering. Then only you can cut and paste DNA molecule. Ligase is just like glue. You have already studied that DNA ligase means it actually seal the break in DNA. That is ligase. Host organism, in this case, E. coli, right? So most of those things I already told you, but quickly I will go through it. Restriction enzymes are a special type of enzyme I didn't discuss yet. Naturally present in some bacteria. Those are restriction endonuclease. Their natural function is destroy bacteriophage DNA in bacterial cell. I should tell you that bacteriophage here, many of you know, those bacteriophages are actually virus. Okay. And their host cell is E. coli, like bacteria. They destroy bacteria. So as a result of that, bacteria, they have some immune system. That means bacteria wants to kill those bacteriophages. How? Because bacteria, they have restriction endonuclease, and this enzyme, they actually cleave bacterial DNA. But you can ask me that then why this restriction endonuclease, this is nothing but some kind of enzyme. Why this enzyme is not cleaving bacterial own DNA? Because bacterial DNA is protected, because most of their cytosine residue, they are methylated, and this enzyme cannot work if the cytosine is methylated. Do not go into that much of details, but only remember that restriction enzymes are used as a defense mechanism in bacteria, so that bacteriophages cannot attack them, right? Now, what is restriction endonuclease? I would like to tell why this name is restriction endonuclease. Restriction means they are restricted. They are sequence specific. They cannot cut DNA here and there. They recognize some particular sequence, and nuclease, you know, any enzyme that cut nucleic acid, nuclease. Endonuclease means they will cut inside the DNA. That means two nucleotide present, and the phosphodiester bond, they will break it. Here you can see endonuclease, right? So that's why they are called endonuclease. So again, I am telling you, I am not going into more and more details, because if you see like previous year question paper, like 3-4 years back, I used to ask some complicated question where you have to use actually, but that kind of question I cannot set in MCQ manner. But the idea is you have to cut your DNA through restriction enzyme, and also you have to cut your plasmid through restriction enzyme, and then finally you have to put your gene of interest in the plasmid. Nowadays, all these restriction enzymes are available commercially. We buy it from good sources, and we can use that. Just next slide, you can see some little bit of example here how it works. So for example, this is one segment of DNA, right? And this portion you are interested. So what you will do? You have to cut this portion actually through some restriction enzyme. So as a result of that, when you cut restriction DNA through restriction enzyme, you will see this kind of sequence you will get. That means you have some single standard overhang like AATTC. Here also TTAA. I'm not going into that details. They actually recognize some specific sequence. So as a result of that, when they cut this sequence, then what will happen, right? They have some AATTC and here also some overhang. How? Then you have to come here again. So for example, after cutting this DNA, what will happen? You will get something like this G, and you have something here. You have something here plus here AATTC, and here you have, sorry, this G is wrong. This G should not be here, okay? And here you have G and something like that. And again, it can be ligated in presence of ligase enzyme. So as a result of that, if you use same enzyme to cut your gene of interest as well as some plasmid, so they will have the matching sequence here, and then you can put your gene of interest in the plasmid, and as a result of that, you will get some plasmid, right? And that contain your gene of interest. By using this restriction enzyme, you can put your gene of interest, in this case, gene A. So that way the plasmid is now ready. So you have to transfer this plasmid into some host cell. So here some frequency of occurrence of restriction site. This is some mathematics basically, that if bases are randomly distributed, what will be the number of restriction site, all those things that you can understand later, no problem. So the ligase is DNA ligase. They seal the double-stranded break, as I told you, that after restriction digestion, then you have to put this digested plasmid, that means clipped plasmid and the clipped DNA together, and then you have to add DNA ligase. In presence of ATP, it will seal the gap, so it will make make the plasmid intact again. So some of the important features of plasmid, as I told you, actually I explained all of those points. That is self-replication. I told you before, it is autonomous. That means bacterial chromosome without bacterial chromosome replication, the plasmid can replicate, can be double in number quickly, so that in presence of antibiotic, it can increase its number. As a result of that, you have already inserted your gene of interest, and your gene of interest will also be increased. Cloning site means here, say for example, you can see here some restriction sites are there. You can put your gene of interest here, okay? And in some engineered vector, engineered plasmid, the cloning site is also engineered. That means you have a lot of different choice of restriction endonuclease sequence, so that you can cleverly use some of those sequence in order to cut the plasmid and put your gene of interest here. Promoter, Soham actually asked me before. Yes, promoter should be there in proper place, and just under the promoter, just downstream of your promoter, you have to clone your gene, or you have to put your gene of interest. And selectable marker means here the antibiotic resistance gene, so that if you increase the antibiotic concentration, then the number of plasmid will increase, and proper size plasmid are little bit small in size, so that it is easy to handle. And apart from bacteria, some other organism we also use in our lab as host, like yeast, plant cell, mammalian cell. You don't need to understand. This is more complex, but yes, all protein, all foreign protein, you cannot make it in bacteria. Sometimes you need to use some other host cell in order to get some recombinant protein. And this is cloning site. That means some plasmid, how it looks like. I have taken from some textbook. And here, this is the cloning method, some schematic diagram, which I already explained, cloning and transformation of a gene. And so as a result of that, you will understand that at the beginning, I told you that insulin, the mature insulin is made up of two polypeptide chain, right? Those are disulfide link. And this is your mature insulin. Then how we will make this in lab? The corresponding sequence of those two polypeptide, so the corresponding DNA sequence, you have to make it. You can synthesize from lab, or you can buy commercially. So this is the DNA that makes the peptide present in insulin, right? So then you can synthesize the another gene that make the another peptide present in insulin, that is the B chain and A chain, and you have to put this genetic information in the plasmid, then you put this plasmid into E. coli host cell, and then make your protein, that is two polypeptides separately you are making in different flasks. After getting those polypeptide, nothing but you have to mix is 1:1 ratio, and finally after some some detailed procedure, you will get this mature insulin. So that way, currently in the lab, we are making huge amount of insulin in our lab, right? So this production of insulin is not that difficult. So for example, I'm just giving you an example. For human use, that needs a lot of quality control. That can be done in a particular setup, like in India, Biocon, which is present in Bangalore. So they are the leader in producing the production unit of insulin, Biocon. Okay, but honestly speaking, even you can make insulin in, for example, if some student they have little bit, not little bit, some thorough background knowledge in DNA, RNA, protein, they can do this project even in one of our lab. This is not very difficult. You have to just clone. You have to just make this thing, and then you have to transform in E. coli, and then you have to make those products. It's not very difficult. But the thing is, you cannot inject those protein definitely without any proper quality control. But making insulin is possible even in our lab setup. Okay, I'm sure that you might have multiple questions. So if you have question, whatever discuss last few minutes, you can ask me. I will address those, and then I will take some break. Then I will discuss the next part.
Excuse me, sir.
Yeah.
Sir, restriction endonuclease cuts at a specific site. So, how is it that our gene of interest is in that specific site?
Yeah. So your question is very good. So there are a lot of restriction enzyme. Normally, restriction enzyme, they recognize 6 nucleotide sequence in general. So you can understand that since we have only 4 nucleotide, there are a lot of chance that here and there in our genome that sequence can be present. You are right. But your question I understood. You are saying that since I am interested on a specific segment of the DNA, this segment only. So how I will ensure that I have that restriction sequence and both the end of the gene of interest, right, Rajesh?
Yes, sir.
Yeah. So that's why what we do, a very good question, very practical question, I would say. But this is like all my research scholars, they used to take care of this every time when they are doing PCR and cloning. So this is gene of interest. Yes, I I understand your point that they don't have particular restriction site which I want to use. In order to do that, while I'm doing the PCR, I'm using some primer, right? You have some primer sitting here, it will amplify, and another primer here, it will amplify DNA. So in the primer sequence itself, the primer will match some portion, like the end of this DNA. So that portion is matching with this end of the DNA. In addition to that, we keep some like excess portion that contain the particular restriction endonucleoside recognizing sequence, some restriction site, some sequence. So as a result of that, after PCR, say after 30 cycle, 35 cycle, when you will have your PCR product, all of those PCR product, they should have that restriction site in this end and also in that end. So as a result of that, now this PCR product, you can digest with some particular restriction enzyme which you use inside the primer, the sequence you have used at the end of this primer, right? So that way you can modify your PCR product by restriction enzyme. So Rajesh, is it clear? And also to all others?
Yes, sir. Sir, so we first extract means our sample of DNA from the human cell, that repository, then we do PCR and all that process. No?
Yeah. During PCR, you have to add this thing. See, to all of you, I should clarify here one thing. We shouldn't use direct DNA because we have intron exon. What we do, we take mRNA. From mRNA to we make cDNA. That means from mRNA through reverse transcription we make DNA, and from that DNA we do PCR. Forget about it. Because if I am not telling you, I am recording the lecture also, this is wrong information. I told you that, but this is not at your level. You can ignore this point. Again, I am telling you, we actually take the mature mRNA from human cell. From the mRNA, there is one enzyme you can transcribe in a reverse way. That means from RNA, you are getting DNA. So as a result of that, you don't have intron, right? That DNA is actually the productive thing that will make the particular, that has specific sequence which will make the protein. That DNA, whatever you are actually generating from your mRNA itself, that DNA you have to again do PCR. And during that PCR, you have to use particular primer that contain a specific restriction endonucleotide that you can design. Whatever plasmid available in your lab, on the basis of that, as well as whatever restriction enzyme you have in the lab, you have to select those things, right? So for example, in my lab, I have around 20 different types of restriction enzyme. I have 10 different types of plasmid. So on the basis of that, I have to cleverly select all those things, and then I have to carry out this reaction. Okay.
Yes, sir.
So that way, you have to put your gene of interest, and then you have to make your product. I hope that most of you, whoever present in the class, you actually have you get some idea how, like, this is like modern biology, I would say. This has been started after 1980 only, because during that time restriction enzyme actually, discovered. Without restriction enzyme, you cannot do all those things. The only thing is restriction enzyme, right? You cannot cut your DNA with any other equipment, right? So as a result of that, after that, it has been, started, but it's steadily increased. Now in every molecular biology lab, we do in a routine basis all these, kind of steps. And now even some more advancements happen. So please take break for 3 minutes, and then I will discuss again. So when you have class at 10:30. So from two different section 1 and section 2, please tell me. So when you have class?
Sir, we have class together from 11.
10:30 or 11?
10:30.
10:30. Okay, so I will be done before 10:30. Okay, I will take around another 30 minutes. But this part is very interesting. Now I will discuss about the cloning of the whole, animal. So for example, you know that Dolly and other things, the basic principle of creating. So here, whatever the cloning I told, this is cloning of gene, right? That means you have one copy of your gene. Now in the bacteria, you have multiple copies of your same gene. But now I will discuss about the cloning of whole animal, the whole organism itself.